Well, thank you very much for the invitation to speak here today. Rather big topic here. We're just going to touch on an overview of human consciousness, and then we might discuss the question of animal consciousness, and then when did consciousness evolve? So not too much to do in a 40-minute lecture. Fortunately, though, we already have the answer, or a pretty good guesstimate of the answer. There was the Francis Crick Memorial Conference in July of 2012. This was held at Cambridge, and they were doing it, all these speakers spoke about the idea of consciousness in humans and non-human animals. And at the end of the conference, they came up with a declaration in which they called the Cambridge Declaration, and I'll just bottom line it for you here, in which they say that the weight of evidence indicates that humans are not unique in possessing the neurological substrates that generate consciousness. Non-human animals, including all mammals and birds, and many other creatures, including octopuses, also possess these neurological substrates. And so all I can try to do for you here is fill in some of the blanks and see how maybe we got to this conclusion. So what kind of consciousness are we talking about? Well, we're not going to be talking about universal consciousness, the oneness with the universe. We're not going to separate into mind and body, or mind, body, and soul. We're just going to limit ourselves here to the idea of a biological process that occurs within the brain, a term sort of called biological realism. And if we want to define what consciousness we're talking about, we'll use Dr. Searle's definition here, that it's basically subjective states that we all have when we're awake, and we know what's going on in our mind, and it continues through the day and it goes away at the end of the day and comes back. Now with anesthesia, I'm an anesthesiologist, we also use this definition. If you don't complain when the surgeon cuts you, it's likely you are unconscious. Now this is an anesthesia machine here, and these are the anesthetic gases in these vaporizers, and it uses some flow of oxygen and air, and it goes through some pipes and tubes and comes out a big breathing circuit, and the gas will be flowing through this tube down into your lungs, and it goes through your body and anesthetizes your brain. And so if I want to actually use the anesthetic gas, not just to anesthetize you, but there's a dose-dependent amount here of how much I turn this knob, how much it affects your brain. If I turn it just a little bit to 0.2, I can study what's happening in your brain when I start to knock out your memory. If I turn it a little higher to like 0.9, I can study what happens in your brain when we make you unconscious. And if I want to see what's happening during surgery, I have to turn it way up to about 2%. So really turn it quite far, and then I can see what's happening in your brain, and it gets suppressed in your spinal cord so that you don't respond. Now, in real anesthesia, what we'll do here is we'll cheat a little bit. We'll turn this down a little bit, and we'll give you a muscle relaxant that'll paralyze every muscle in your body, and you can't move. And if we do this wrong, you can look like you're asleep, but you could actually be awake under there. And this is a very bad experience for you, and it also leads to a number of bad movies. So what basic facts do we know about the brain that we can use to sort of rein in our criteria here a little bit? Well, this is from Seth Barr's and Edelman's paper on looking at some basic brain facts. Now, they went through a list of 14. We'll just go through 3 here. But basically, you have an electroencephalogram, the EEG activity. And when you're awake, it's in a irregular, high frequency, low amplitude signal. When you become unconscious, that regularizes and gets much higher amplitude. This is from a recent review from George Mashour's group showing some of the criteria we might use as we increase the dose of anesthesia to put you asleep during anesthesia or knock you out. And here you see that EEG is that fast, high-frequency stuff. And as we increase the dose here, the EEG starts to regularize and starts to have high-frequency, low waves. And if we give you enough anesthesia, we can actually flatten all the electrical activity in your brain. And so we can be pretty sure with a biological approach that if your brain is completely flatlined, there's probably no consciousness occurring at that time. So what does it look like when we put you under anesthesia? This is a study we did a few years ago, and it's one subject using PET glucose scanning, and the subject's being studied in the awake state. We brought him back later, studied him when he was sedate, then studied him when he was unconscious, and then under surgical levels of anesthesia. And you can see that as you increase the dose of anesthesia across your whole brain, the brain activity decreases. One of the questions then is, what's different between being here and still conscious and being here and unconscious? That's one of the things we want to look into. So we also know from basic brain facts that there's neuroanatomic structures. There's some that are more important than others for allowing you to be in a conscious state. You need your cortex and your thalamus probably interacting with recurrent activity. And in humans, it seems like the cortex is where the specific contents of consciousness might be held. The brainstem seems to modulate your arousal level. So the idea here is that a cortical lesion, and we know this from much neurology, if you have a stroke in your left frontal lesion, you might not be able to speak. A stroke in the back of your left side, you might not be able to understand words. So according to this idea then, lesions out in the cortex change the contents of your consciousness, but not so much the level. But lesions down in the brainstem here could certainly decrease your level of consciousness completely and wipe it all out. And this is sort of a graph showing where different arousal structures are in the brain and how they feed forward to different areas to wake up your cortex. And then the other idea is that you need widespread brain activity to allow you to have consciousness around the different parts of the brain. And that if you have unconsciousness activation in a very small area, it doesn't necessarily lead to consciousness. And so Victor Lamb put this together this way. This is a brain on the side, and the pointer's about to go out here. But basically, when a percept comes in, this is like the talk we just saw, the idea is that the information comes into the various areas of the feature detectors that put the image together. There's sort of a sweep of information that kind of maybe binds all this together. And then once that happens, the information is almost becoming conscious. And then, sorry, going backwards there. And then there's another sweep that comes from the frontal, that top-down point that we were just talking about, to then integrate with all that and cause a conscious percept. And so this is a little movie showing how this sort of works in real time. This is from Robert Thomas' lab in the Mind Institute, and it's not really a study of consciousness per se, but it shows kind of how this works. This is a MEG study, and the task for the subject was just to choose one hand, one thumb or the other, when a stimulus showed up on the screen. And you'll see here the information show up first in sort of the parietal visual, or the visual areas, and then it'll sort of spread out in the what and where pathways. It'll come forward to the somatosensory cortex. There'll be sort of a large integration of something happening, and that feeds forward to the frontal cortex, and then maybe there's some secondary emotional component to what happens. And so I'll play this through. This goes through in slow motion here. Oops, sorry. And so you're seeing the MEG information of processing a single visual stimulus going across the brain. Yep. I'll show you that again. And you can get a sense of the feedback and the feed forward that we were talking about in the last lecture if you kind of concentrate on sort of these areas over here. And this is slowed down quite a bit. We actually can speed that up to approximate real time, and it looks something like this. So that's your brain processing a simple visual stimulus. Now, you can imagine that at the same time, you're also hearing things and feeling things, and so as your brain is doing its conscious thing, your brain is essentially on fire with all this activity across the whole brain. So how do we then start to be able to study what of that firing is related to consciousness? And Stan Dehane's group has done a task where he's tried to look at the level of processing in order for something to become conscious and what's changing in your brain when that happens. So what he does is he takes a little target stimulus, like the letter 6 here, and then he uses a variable delay and then masks out that. And then depending on how long that delay is, you either will or won't be conscious of having seen the number 6. Now you can objectively report whether the number was greater than 5 or less than 5. And then you can subjectively tell us, well, did you see it or not? And what we find is you change that delay period, there's a threshold here for when the object turns into a conscious perception. And using ERP recordings, what they find is that, kind of like the movie I just showed you, here in the back of the brain, down here in the back of the brain, there's a sort of a double hump according to whether or not the thing is actually perceived. And there's an initial early wave and then a second wave, and it's thought that that second wave is due to what's happening in the frontal cortex feeding back and generating that integrated information leading to that conscious percept. Another way to look at it, they also did a study where they actually put electrodes on the surface of the brain in people who had epilepsy, and they were used a mask paradigm again. And in this time, you can actually see the size of the signal changing whether or not the percept was conscious or not. And so we'll run through this, and you'll see now the EEG underlying signal is getting larger in microvolts, whether or not they consciously perceived it. And you see that it doesn't get all that big in the various areas that it might travel through. But on the other hand, when the percept became conscious, some changes actually occurred, and the size of the stimulus across the visual areas gets larger. There's sort of that recurrent processing right within the area, and then there may be even some forward, frontal to parietal feedback effect occurring at the same time. So what this is suggesting, or what it shows, is there's a number of sort of neural correlates of what you need to get a conscious percept happening. And there's other ways to encode the information, not necessarily just the magnitude of the information in a certain area, but also the timing and how it's interrelating to other areas of the brain. And this leads us to Giulio Tonani's information integration theory of consciousness, which is an attempt to get over the hump from objective and subjective and actually turn what's happening into the brain into a conscious experience. And with this theory, recently updated, it essentially says that the neural substrate of a conscious experience is a complex of neuronal groups within the thalamocortical system, which has two properties, and that it behaves as a single integrated entity, and it also has a large repertoire of different firing states or firing patterns, so the occurrence of any particular state is highly informative. And the amount of information that comes from that isn't so much what's firing when, but thinking of your whole brain as a unified system, it's what isn't happening that's also is the amount of information that's being reduced. And so there's a lot of math that can be used to describe how this works, and rather than using the math, I'll show you visually. So this is Henry Markham's picture of his blue brain supercomputer. And what they're doing, this is a guy sitting over here, and what they're doing with this computer with 8,000 CPUs is they're trying to simulate the functioning of this amount of tissue, which is one cortical mini-column in a rat brain. And so you can get a sense of that there really is a massive amount of information that you're integrating in order to make this conscious percept. So we wondered, well, why does anesthesia make consciousness go away? And using the integrated information idea, there's a couple of ways you can see if that might happen. There can be decreased connectivity between the little modules that need to talk to each other, or there can be a total loss of the informational content, and everything starts oscillating at the same pattern. And Giulio's group did this experiment where they're looking at functional connectivity in people under both sleep and midazolam anesthesia. And what they did here is they took a TMS pulse and put a single pulse into the brain and then recorded with the EEG to see how that information traveled around the brain, or if the brain was in a state where the information could travel. And what they found in the conscious state is when they made the pulse just over the premotor cortex, the pulse was able to actually travel all over from front to back and side to side, and taking a number of milliseconds, over 300 milliseconds to do that, suggesting that the brain in that state is in a place where it can integrate the information. In the unconscious state, when the subject was on midazolam anesthesia, a single pulse just made one pulse in the area where the probe was, but it didn't spread around, so it wasn't able to actually have all that information be able to integrate. And so taking this to the clinical side, George Mashour's group has looked at how information transfer is happening in the brain as you're coming under anesthesia. And what happens here is this is looking at symbolic entropy, transfer entropy, and in the baseline state there's this normal front to back balance that you're getting this feedback from the front onto the back of your head. And as you go and increase the amount of anesthesia, it starts to decrease that balance, and then when you're anesthetized, there's no longer this front to back entropy transfer. When you start recover, that comes back. And again, there's a front to back feedback to everything. And he's shown this now with a number of different anesthetics, sevoflurane, propofol, and ketamine. And ketamine is unusual because it doesn't do that whole major decrease of brain metabolism like the other ones does. So it's just showing how that information capacity gets decreased with the increasing dose of anesthesia. So if we take these basic brain facts and look through sort of the evolution of animal consciousness, we're looking for things that have, of course, EEG activity, maybe a cortex and a thalamus-like structure for recurrent processing, and then widespread brain activity for sort of this global neuronal workspace implementation, as well as a high capacity for integrated information. And now this capacity for information integration can be quantified, and depending on how complex the system is, you can get sort of a measure called the phi measure that gives you a number that kind of gives you a sense of how much capacity that system might have. And so now we can discuss some alternatives regarding animal consciousness. So perhaps animals have no conscious mind, they only react to stimuli by desires and instinct. Well, this seems much more possible when we had the idea of the scala naturale, where we had evolution proceeding from a sort of a simple organism and adding on new structures and new substrate and different tissue as you go up the scale. And you might propose there's some line at which, well, you have to have certain structures in order to have consciousness. And in fact, one line used to be thought of, well, maybe at the level of birds. Well, there's recent good evidence that, in fact, the bird brains are not organized as we thought from the old morphology studies, but actually the bird cortex is a homologue of the mammalian neocortex. And we know that how birds can perform is actually they have quite advanced behaviors. They have causal reasoning, imagination, they can use tools. And so the idea of the birds being something of a lesser species doesn't really hold up. And in fact, the more modern theory is that as you evolved, most of these structures probably were still in there for every creature, but they just adapted to different niches, and different bits of them got larger. Well, so maybe animals have limited primary consciousness, and their brains are evolved just to their basic survival needs. Well, there's some good data with the test called the mirror self-recognition test that animals, some animals at least, may have something more than just limited consciousness. You certainly know when with your dog, when you come home, he certainly seems to recognize you. And Gordon Gallup did this study where he studied chimpanzees back in 1970, exposed them to a mirror, let them get used to seeing the reflection in a mirror, and then anesthetized them and put a mark on their head, and then showed them the mirror again. So if the monkey was able, or the chimpanzee was able to have a sense of itself and recognize who that was in the mirror, you would expect that it would sort of touch and play with the mark. And that's in fact exactly what they found. And in fact, the ability to play with that mark or see that mark and recognize yourself is limited essentially to the great apes, and that evolves in humans around the age of 2 years old. And then recent studies with dolphins and elephants have shown that they can also do this test, and most recently with magpies. So there's a sense that those brains are capable of generating at least a self-image and some sense of self-awareness. So what about then the idea that animals have similar consciousness to us, with capacity proportional to the size and complexity of their brain? As Darwin said, a difference of degree but not kind. And Darwin was sort of understood the idea that the emotional nature of much of our life sort of guides our conscious behavior. And if you want to be a mind reader, it's actually not so hard to do. Here you can see this is a cat that's kind of a happy cat, and here's the cat you wouldn't want to approach. Now, even in humans, our facial muscles are innervated by down deep in the brainstem, all the same sort of neuroanatomy that applies across all the different species. So if you want to read someone else's facial expression, it tells you about something about their state of mind. And so where is this sort of coming from? Well, Derek Denton has this idea, or came up with this hypothesis, that the primal emotions, your need for when you're thirsty or when you're hungry, when you have air hunger, where that might be coming from is down deep in your brain stem. And that perhaps the evolution towards consciousness sort of grew up from this idea of interoreceptors, your basic biology need to maintain homeostasis, rather than so much of a top-down process of exteroreceptors or scene creation, and then there was some consciousness. So perhaps that there's this level of consciousness in everything. And so the question might be, how primitive do we have to go to find a creature that's capable of making choices? And this is the lamprey here, and it's evolved about 560 million years ago. And you might recognize that picture there from Pirates of the Caribbean. That's what Captain Jack said as he was about to get eaten. And so recent neurophysiology work and neurochemistry work from Stephen Jones' lab has shown that even the simple lamprey, as we were talking earlier, has a basal ganglia that is capable of both action selection and not in doing a particular action. So it is evolved from a very early age in evolutionary terms. It's evolved this ability to make choices. And this underlying neurophysiology, this underlying structure, has evolved at the earliest radiation of the vertebrates, and suggesting that underlying all of us is this very basic choice mechanism that we all have. And so in some of our studies at the center here, we're looking at how anesthetics affect the processing and memory of emotional pictures and what happens with emotional memory. And in our studies here, we had a simple study where we were looking with fMRI and people seeing a series of pictures while they're in a scanner, and then from there seeing which they remember later. Now, I'm not going to show you the memory bits just now, but all their task is to look at a picture and say whether the picture was emotionally arousing or not. And we didn't define it too much for them. And so when you do that, and you look at the contrast between the pictures that they found arousing versus pictures not arousing, what we find that activates is the amygdala bilaterally with the brainstem, and the whole brainstem and thalamic system, along with that anterior cingulate, the exact circuit we were talking about in the last lecture. So again, suggesting that at a very basal level, the circuits associated with your basic consciousness are very deep down in your primitive brainstem. And then this is a study we've done where we were looking at people actually coming out of anesthesia, and we wanted to know what was happening that was different in their brain between when they started, when they were sedate, when they lost consciousness, and then when their consciousness finally came back. And it turns out, clinically, when you wake someone up from anesthesia, we know when you're about to wake up because we're able to see the very basic brainstem reflexes coming back online. We'll see your eyes, which when you're unconscious are pointing in opposite directions, they'll start to come back together. You'll start to swallow. You'll start to do, your breathing will become regular. And when we looked at that with PET scanner, this is data from the Turku PET Center in Finland, and we analyzed it here. We see what's turning back on is, again, your brainstem, your thalamus, and that emotional part of the limbic system, the anterior cingulate. Not so much the cortex coming back on. In fact, this fits with our idea of what happens when you wake up in the recovery room. It takes quite a long time for you to actually be sort of aware of where you are and what's going on. So it's as if the cortical parts have to come back on a little bit later. And so what happens then with other anesthetics and other ways of being anesthetized, in terms of, so what we see with the number of different anesthetics, if we subtract the awake state from the anesthetized state and just do the subtraction image, and this is a number of studies across the world from different labs using different anesthetic techniques, and even though they're showing different pictures of different colors here, in each case, the colors represent where there's the most change from conscious to unconscious. And it seems to be a area in the thalamus and brainstem that seems to be turning off the most in this switch from conscious to unconscious. So what might be going on here in a sort of a neuronal level that's making this sort of switching mechanism? Well, from Steryad's work and others, there's the idea that the thalamocortical cells, in fact, the whole cortex and reticular thalamus, it tends to oscillate in two separate modes. It has sort of a burst firing mode and a tonic firing mode. And the different modes are correlated with whether you're in a conscious or unconscious state. And if you apply any sort of arousal substance in a petri dish to simply a cortical neuron, it'll switch its mode of firing. So these basic ionic pathways that have this happening are very much conserved throughout evolution. And what we see here from Rodolfo Linus and Steryad's review is that even in fish, frog, turtle, birds, this switching mechanism seems to be available across all of evolution. And so one other thing to show you here then is, so what happens, what's different in that brain between when you were conscious, this is going back to the Turku data here, between being conscious and unconscious, is we take the activity in the parietal lobe and we correlate it to what's changed in the brain when you wake up. And what we find is that there's a reconnection of the parietal lobe with that frontal anterior cingulate lobe. And so you've reactivated the thalamus, reconnected your cortical things, and then you're essentially back into your conscious state. And so how does this work then in sort of an animal model? So we wondered if this change in thalamic functioning is really causal or has anything to do with your changing level of consciousness. And so we tried injecting arousal substances into the brains of animals. And let me show you this little thing here. So we're basically trying to see what will happen. So here's a rat in a box, and the box is full of 1.4% sevoflurane. And in there we've got a little cannula that's going into this thalamus. And we didn't know exactly where we were targeting in the thalamus. We just knew we probably wanted to target the non-specific thalamus. And when we give us about a half a microliter of nicotine just directly into his thalamus, even though the whole rest of him is still in a box full of sevoflurane, we get a response like this. So the little guy gets up and starts to move around. So suggesting that that change in thalamic functioning, that gating between having arousal and not having arousal is a fundamental thing that you need, evolutionarily conserved all the way down to the lamprey. This is where we actually had to put infusions in order to get this to work, and the main target turned out to be the central medial thalamus. And this is kind of interesting in the rat, because at the same time we were doing this work, Nico Schiff found in humans that if you stimulate that area in someone who's in a coma, or actually in a vegetative state, you can actually enhance their arousal and get them to improve their condition. And so all that takes us kind of back to where we started, and the essence here that the absence of a neocortex does not appear to preclude an organism from experience affective states. Convergent evidence indicates that non-human animals have the neuroanatomical substrate, neurochemical, and neurophysical substrates of conscious states, along with the capacity to exhibit intentional behaviors. And so again, the weight suggests that most of the creatures, invertebrates, have the ability to have consciousness. And I want to thank George Mashur for his help in how to put together this lecture, and the group from the Turkey Pet Center for their excellent data. And thank you very much.